It's super interesting, and it is great that you are connecting all these molecular dots. And when you look at those dots, the data points that you're looking at, right? Because a lot of people, I think, when they think about dots, they're thinking about, oh, what lab tests are you running and what dots are those? But it goes beyond just the, let's say, functional labs and the normal labs. You're looking at genomic, proteomic, metabolomic data. And that's where it gets really interesting, but also somewhat confusing to the layman that's like, what is that, right? What is that?
Yeah.
My triglyceride reading like that I'm used to just seeing and all this and that. It's further molecular dots. Can you talk about the differences, maybe just in those three? I mean, I've heard those terms. I know how some are using. I'd love to know how you're using those data points and connecting the dots.
Yeah, happy to. So, essentially, we just went over genetics, right, and genomics and varying levels of clinical application. One thing I did neglect that's leading the charge of precision medicine is pharmacogenomics and looking at, who's going to respond to what drug based on what and how and dosing and so forth. And that has to deal with like cytochrome P450s in the liver and the rate of metabolism. Some people are fast metabolizers, slow metabolizers, and that influences, your receptivity to intervention or that has those metabolic kind of pathways. But speaking forward, going back to the central dogma of biology, which is DNA into RNA, RNA into protein, essentially DNA codes for genes and genes code for proteins, right? And then proteins are, these structures in the body that give rise to structure and function. And then all of a sudden you have proteins that give rise to enzymes, and enzymes give rise to metabolism, and metabolism gives rise to physiological function. So it's building off on that narrative where, there's various levels of proteomic signaling based on, just the signals and inputs that we're feeding our body. So for example, if you eat a standard American diet and it's just loaded in like high fat processed oils and all the bad stuff, right? Or glyphosate or all these other environmental toxicants that we're exposed to, what's going to happen? You're going to drive up these inflammatory signals like nuclear factor kappa beta, right? And you're going to have all these other inflammatory signals that are upregulated on a transcriptomic level, that's coding for these proteins that are pro-inflammatory, right? So I do believe from a proteomic standpoint that, and I've spoken to some of the top researchers around the world on this. From a proteomic standpoint, you can measure all these freaking proteins in the world. So you could just measure so much, right?
Right.
Hundreds, thousands proteins. So what? That's great. Tell me what's the end? What am I going to do about that? Right? Because at the end of the day, there's going to be, I think, four key kind of phenotypes associated with perturbations within the homeodynamic regulation. And that's going to be inflammation, oxidation, ischemia, and immune dysregulation, right? That's what I see clinically all friggin' day, right?
Mhm.
Is oxidation, inflammation, ischemia, and immune dysregulation. And if there's another kind of proteomic deviation, I would love to hear about it and unpack that further. But for me, that's what I see clinically day in and day out. So proteins, we can measure all sorts of different proteomics within the system. And that's going a little bit deeper. And you can even do this with LabCorp and Quest by going into the lipidology and looking at LPA and LPPLA2, ApoB, fraction size and all that fun stuff from that perspective. You could look at homocysteine and, as an indirect reflection of the MTHFR gene and how that influences, folic acid utilization. You could look at, there's what is it, the beta amyloid 42/40 ratio in regards to accumulation of some of these misfolded proteins in the system. Right. So there are different levels of proteomics that you could go into, right, to get a better appreciation of what's going on and translating this information from genome to phenome. So that's the proteomic kind of dynamics that I do clinically. And then in regards to metabolomics, well, again, proteins give rise to enzymes, enzymes give rise to metabolism, metabolism gives rise to function. So the metabolomics of this is now looking at, deeper kind of complexes of where we could identify any perturbations within the system or the cellular function and physiology due to maybe toxicity or deficiencies, right? So identifying any of these potential toxicants that interfere with these enzymes that are involved with like metabolism and function, cellular respiration, whatever it is, we could identify that, right? And I actually have tracked it both on a transcriptomic level, right, and a metabolomic level, and they're telling us the same thing. And it
Mm.
goes back to, basically saying, hey, there are these various factors in our environment that influence our ability to have normal function, right? So in my one of my practices through the university, I work with Dr. Andrew Heyman, who's the director at A4M, really big researcher in the field of chronic inflammatory response syndrome, CIRS. And in our practice, we use a test called Genie, and Genie is a transcriptomic test. And so if you were to imagine your DNA being the instruments to your own personal symphony orchestra, the transcriptome is measuring the melody coming off that gene, right? So are certain instruments playing too loud and upregulated, or certain instruments playing too low and downregulated, right? And what we end up ultimately seeing is this molecular signature that's very consistent with, various levels of environmental stressors, whether it's infection, Lyme disease, co-infection, chronic viral issues. We see the molecular signature of cell danger response based on the research of Dr. Robert Navinow. And essentially, when your cells perceive danger, it responds by conserving energy and downregulates your mitochondrial large and small subunits and translocase and ATP synthase. And that's why you feel like shit, right? Because those genes your cells are perceiving stress. It's downregulating. It's taking on this defensive posturing, and it's conserving energy, right? Because of that threat in the environment, right?
Yeah.
We also do research around the FKBP5 gene, which is associated with PTSD, and we see that in a lot of individuals. So it's very interesting looking at things from that transcriptomic level. And then, if you do a metabolomic testing, right, you're able to see a very similar pattern of mitochondrial dysfunction due to CDR, cell danger response, or biotoxin-related illness, or infection, or, your immune system can't keep up with the pace of immunological surveillance because it's so inundated with noise, coming in from all the different environmental threats, whatever that might be, right? Environmentally. So there's just so many different factors there that we're able to kind of pinpoint, that influences your phenotype. And when I had a call with the NIH the other day, the director of exposomics, and I told him what we're doing with the American board, he was like, holy shit, this is incredible. Like, we want to support you. And
Yeah.
because it is the future of medicine.
Well, it's so many more dots and data points that answer the question, why do I feel like shit, that we're just missing a lot within conventional medicine of guessing and just putting a diagnosis to it without understanding the why. And you don't stop at just those data points. I mean, you're looking at dark field microscopy. I see that in the background. You're looking at various ways to start to understand the why I feel like shit. And when you get that answer, treatments becomes very precise and personalized.